Welcome to part 2 of the Introduction to Endocrinology. Continuing, we're going to be talking about these feedback mechanisms. Like I said before in the previous video, this is going to be huge. This is where a lot of your general, basic foundation knowledge is going to come from understanding this material in this specific way. This is going to give you, what is released where, what does it affect, what does it act on, how is it regulated, how is it controlled. Test question central for professors. So negative feedback, this is when the endocrine cell releases the hormone, it goes to the target cell. Target cell, does its function, does whatever it needs to do, and that causes a negative feedback. It causes it causes the endocrine cell to then stop releasing the hormone. Positive feedback, which is much rarer. There's only one or two examples that we will go over in the course of this class, where we actually talk about a positive feedback loop. So much more rare. Most of the things will be negative feedback, but basically the endocrine cell releases a hormone, acts on a target cell. Target cell is then going to stimulate the endocrine cell to release more of the hormone. So negative feedback decreases the size of the stimulus, and this is the majority. This increases the size of the stimulus when bigger response is needed, when there's a definite end point. Specifically, the one we talk about with positive feedback is going to be Make sure I spell everything right. Positive is the oxytocin. So oxytocin is stimulated due to pressure on the cervix from the baby's head, and causes the pituitary and everything to release oxytocin, which increases the contractions, which causes the contractions to be stronger, which puts more pressure on the cervix, which then causes the release of more oxytocin, and that's the positive feedback loop. This is always, 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 always a test question. Every single time. Every single time. Okay. Here are an example of negative feedback, where the connection of the hypothalamus to the anterior pituitary. Anterior pituitary releases a hormone that has negative feedback on that. Anterior pituitary goes to a different peripheral endocrine gland, continues on to target cells, and that also feeds back and inhibits the release of more of the hormone. This is what the negative feedback loops are going to look like. And again, remember, the reason why this works is because all of these hormones are traveling through the blood. Remember, they're systemic and they're slower acting. So by the time, the hormone is released, it's cycled through the blood, and each of these different glands is going to have a receptor in it checking the percentages of our blood, kind of checking the concentration of these specific hormones in the blood to make sure that they're appropriate. Lots of stuff can go wrong in here as well, so this is where you will learn about a lot of different conditions, and I'm going to try and touch on those conditions to prepare you as best as I can for systems pathology in Try 4, because that's going to be that's one of the beastly classes. Is a really, really big class, a ton of information, and a lot of this is going to be the first big chunk of that course, so make sure we pay attention a lot to the endocrine system. Here's giving an example of all this. Another example of negative feedback. So during the menstrual cycle after ovulation, the ovary begins to secrete progesterone in response to luteinizing hormone. Progesterone inhibits the release of the GnRH. That's the gonadotropic releasing hormone. Double check. Yep, the gonadotropin releasing hormone from the hypothalamus, and then LH, luteinizing hormone, from the anterior pituitary. And then once those decrease, progesterone release will also decrease. So it's all connected within that loop. So decreased gonadotropin releasing hormone reduces luteinizing hormone secretions, and so the overall levels of the GnRH and LH levels in the blood decrease. This is that oxytocin loop that I told you about. Again, like I said, 100% on almost every test, because they're going to test you on positive feedback, and there's literally, this is the only way to do it, is through this example. Hormone secretions, you have chronic or acute or cyclic. Chronic meaning that it's a maintenance of relatively constant concentration of the hormone. So thyroid hormone is a good example, where you constantly have to have the thyroid hormone to maintain consistently. Epinephrine is a response to stress. Cyclic, the female reproductive hormones, which we'll cover later on in the class. It's not for a while that we talk about those. We do need to know and learn kind of the groupings now, even though we're not talking about all of these now. We need to know which ones are steroid, which ones are protein, which ones are amine hormones, so that we don't have to worry about that later on. So know all of these now on these hormones, but understand that most of the information on these specific hormones and that kind of stuff is not talked about or discussed until later on in the course. If we pause and we think and kind of compare some of these other parts, so other hormone secretions, like when we're growing, growth hormone is going to be pretty consistent, more on the chronic level. Acute, think insulin, glucagon, those ones are going to be really, up and down, up and down, up and down, depending on our diet and throughout the day. Methods by which feedback control is exerted. We can have neuronal control, humoral, meaning relating to body fluids, such as the blood concentration, and then hormonal, the actual hormones themselves. Humoral control, the blood levels. So this is like glucose and insulin. When the blood glucose levels increase, glucose diffuses from the blood and stimulates the pancreas to release insulin, which diffuses into the blood. So it's controlled by the blood levels of blood concentrations of specific substances. And like these examples are the ones that they'll most often test you on. Nervous system, so sympathetic nervous system, epinephrine and norepinephrine, prepare the body for those stressful signals. And when we remove that stress, we get less of the epinephrine released. So that's thanks to the nervous system. Hormonal regulation, this is like our classic negative feedback loop that we'll see a bunch of. Hormones themselves, the way that they travel, we said they use the blood. 2 different ways that they can go through the blood. They're either going to go in their unbound form, or they're bound to plasma protein. Think like albumin. It's one of the big ones for binding proteins and everything in the blood. Both of these forms can diffuse through the capillaries into the interstitial fluid and bind to the target cells. So just reiterating the point, hey, it's through the blood that these travel, either unbound or bound to plasma protein. Most commonly, a majority of them are bound to plasma protein, is what most often we will see. But again, we'll go hormone by hormone. If there's anything else specific, then we'll mention that. Water-soluble hormones will be free in the blood as it can diffuse into the plasma. So like these ones, this is the exception. These are going to be more in the unbound form. And compared to, like the steroids, the lipids, the other ones that are going to need to be bound in order to continue to process. Lipid-soluble hormones, the steroids, they absolutely have to have the transporters. Another little note, the half-life of lipid-soluble hormones is a lot longer than the water soluble, because water soluble hormones, again, because they are not bound to anything, they're just free floating around. It's more likely, that they get stuck, that they get trapped, that they get destroyed, they get acted on. Something happens that they break down, versus the lipid soluble hormones are bound to something and they have a transporter, so they don't break down quite as quickly. Here's just little steroid hormones and thyroid hormones. You have from the endocrine cell, it releases a free hormone, can break down, or it goes up, gets bound, or binds to the receptor, and you get the biological effects. The free hormones are susceptible to breakdown. Here are some of those specific transport proteins for those. These aren't tested on a bunch, but it's still good to know and kind of correlate these. So like the corticosteroid, cortisol, aldosterone, thyroxine binding globulin, thyroxine, triiodothyronine, and then the sex hormones, testosterone, estrogen. So it is relatively straightforward as far as like the naming goes. You should be able to recognize pretty easily which one it goes with what. Some of the non-specific, albumin in the blood, that's the big one, the most common protein produced by the liver. Most steroids, thyroxine, triiodothyronine, geez. And we also have transthyretin, which is thyroxine in some of the steroids. Blood levels of hormones are determined by the rate of secretion and the rate of removal from the bloodstream. So again, it's just constantly checking how much is in the blood and how much is comparing it to how much there should be, and then determines how much more it needs to secrete. This is the rate of removal from the bloodstream. We call it clearance rate, the rate of disappearance over the concentration of the hormone. That is expressed as half-life. So it's half-life is referring to the amount of time it takes for half of the hormone to be broken down. So if we think like outside chemicals, other things like caffeine has a half-life of about 12 to 24 hours. So in any 24-hour period, when you drink caffeine, within the next 24 hours, if you drink, so let's say you drink 200 milligrams of caffeine, you have a Monster energy drink. The next day you have, this is 24 hours later, at the same time you drink another Monster energy drink. Within that 24-hour period, 100 milligrams of the caffeine has broken down. Now you drink another 200, now you have 300 milligrams of caffeine in your system. And then by the day after that, it's down to 100. Now then you would have 150 in your system. You drink another 200. So this is why it's important to recognize, understand half-life and how it works, not just with the hormones themselves, but also with other medications, other supplements, other products and stuff that you're taking, to make sure that you're doing so safely within your body. So mechanisms of removal, getting broken down, they bind to the target cells, or the liver and the bile and the kidneys. The liver can take them out, can filter it out. The kidneys can filter it out into the urine. So those are the different ways. Here's just some of the data, kind of what the half-life and the clearance rates are. These are super specific numbers. Again, most of the time, these are not tested on, just because of the super nitpicky specificity of these guys isn't typically what a teacher wants you to know. They're not the most important as far as what you could remember. So if your professor wants you to know the nitty-gritty little details of this, then go ahead and do that, but otherwise don't worry too much about it. Mechanism of action of the hormones. We've already touched on this. Hormones will bind to receptors on cell membrane, like the proteins, glycoproteins, polypeptides, epinephrine and norepinephrine, G protein coupled receptors, all that kind of good stuff. Or they can go right through, if they're lipid soluble, those some of those hormones, can go right through and bind on intracellular receptors. They can also go and they can bind onto nuclear receptors. Remember that from biochemistry? The nuclear receptors inside of the nucleus, so these can all come in and act directly on the inside. This part, the water soluble versus lipid soluble, should be a little bit of review for you guys. Regulation of the number and sensitivity of receptors. We talked about this as well a little bit briefly in biochemistry. We have downregulation and upregulation of receptors. So so the number of receptors is not going to be constant, but can change depending on what they're exposed to and what the body requires. So downregulation of receptors, decreased number of receptors on the outside of the cell, so it becomes less sensitive to effects of hormones. Diabetes mellitus type 2. This is the issue. Essentially, with diabetes mellitus 2, the receptors are bombarded constantly with insulin because you constantly have high blood sugar, so it's just insulin all the time. And because there's so much, the body down-regulates the receptors so that there are less of them. And that's when we get issues with diabetes mellitus 2, because then all of a sudden our insulin sensitivity is way, way down. Our body can't process the sugar and everything that we're consuming, and as a result, blood sugar is way too high. There's way too much glucose in the blood, and then that starts to cause damage. That causes damage of the vessel walls. It can cause damage of the nerves. It can cause damage of organs, all that kind of stuff. It can cause brain damage, depending on the level of sugar and kind of where it's at. Next, you get upregulation of receptors, so it increases the number of receptors on the outside of the cell. You become more sensitive to effects of the hormones. So actually, some of the medications that you could take does upregulation for diabetes mellitus, but as well you can become more sensitive to hormones like insulin by giving the body that break, reducing the number of carbohydrates, reducing the sugar that you're consuming, losing weight, exercising, all that kind of good stuff can boost your insulin sensitivity. Yeah, so when hormones bind to a receptor in the target tissue, the receptor molecules are degraded and replaced on a regular basis. Downregulation. So because when they're binding constantly, the receptors themselves get broken down. If you're binding constantly to them, you have less receptors. It takes longer, to build them, or it takes more effort to constantly be rebuilding them, so your body isn't able to keep up. All through here, then again, there's upregulation. Intracellular signaling. Recognize all of this. Our G protein coupled receptors. They can be produced because of the G protein activation. They regulate the intracellular enzyme activities. I'm not going to go deep into this again. If you want to remind yourself about this, the cascade effect, all this kind of stuff, go back to Biochem 1 and review all this. Some of the second messengers, cyclic GMP, cyclic AMP, calcium, IP3, that is iodine phosphate. IP3 is inositol triphosphate. Then IP3 equals inositol triphosphate. DAG is diacyl glycerol. NO is nitric oxide. So intracellular receptors, Again, should be review which ones, all these mineral, that's spelled weird. Mineralocorticoids, glucocorticoids, thyroid hormones, vitamin D, all of these guys have the intracellular receptors, which then go and they cause transcription directly on transcription to make stuff. This is a good example. Aldosterone coming in here. This is a good example of a nuclear receptor. I should say this is intracellular. Sorry, this is intracellular, which then goes in and acts. This is not nuclear. The steroid and thyroid are our nuclear. They go and they bind directly onto everything in there. More definitions: synergistic, permissive, antagonistic. So there can be hormones that work together to have a greater overall effect. So they boost the effect of one another. Permissive, a small amount of one hormone helps another hormone to have its full effect. The good one to think about this, like vitamin D and calcitriol. I'm pretty sure that's the name of me. I'm trying to remember the. Yeah, vitamin D calcitriol is an example of that. Antagonists, they work against each other, so one increases the response, the other decreases the response to maintain that homeostasis. Insulin and glucagon. The more on the agonists and antagonists. The agonist, a hormone agonist is an analog or like something that goes along with it. That binds to the receptor and mimics the biological action of the endogenous hormone. A hormone antagonist binds receptors but does not provoke the normal biological response, so it stops the hormone from binding. So this is, again, just talking about like the competitive inhibition that we talked about way back in biochemistry. Just some important points you can pause to read if you want to read those. Here's a lot of the next little chunk is going to be the overall review, the things to make sure that you take a look at. So those general ideas, the ones that have specific, some hormones will have only one specific effect in target cell. They can have multiple effects on multiple target cells, and sometimes those effects involve multiple hormones. More just important points, again, pause to read. More the functions of the endocrine system, all of these guys. Pause to read: water balance, immune system, heart rate, blood pressure, blood glucose, nutrients, reproductive functions, uterine contractions, and milk release. Endocrine dysfunction, we can have hyperfunction or hypofunction. These are going to be where all of our conditions come from. So most often they're going to be written as hyper whatever or hypo whatever. So like hyperthyroidism, hypothyroidism, meaning too much thyroid hormone, not enough thyroid hormone. And we will go into kind of the causes of those and what the issues are and how you can actually tell where the issue is based on looking at the levels of the hormones in the blood. This class is going to tie heavily into lab diagnosis. If you are in the blended program, you will take lab di, I believe, next trimester in Tri 4. If you are in the standard course, you will be taking lab di in Tri 5. Just some of the causes of endocrine dysfunction. You can have tumors are a big cause, autoimmune conditions, iatrogenic, meaning like medically caused. So like you can have issues like due to other medications or other conditions, infectious or inflammatory, or activating receptor mutations. So these will go into all of these a little bit in this course, more so in systems pathology in Tri 4. Here's again a bunch of the causes of the hypo function. At this point in time, what I would do is all of these guys here, all these different conditions, we're going to see these. Put these all in a chart somewhere, all of these guys, because these are all going to come back, and they're going to have a bunch of different conditions, all that kind of stuff. You're going to want to make like a nice, pretty master chart with all of the hypo functions, or just get my notes for systems pathology. If you pick those up, all of those are going to be in there. All of these conditions, you're going to see all of this stuff in the Tri 4 systems pathology. And there we go. That is kind of our first introduction to endocrinology. I think the next couple of videos, what I'm going to do is I'm going to try and go through these beforehand a little bit more and kind of mess with things and make these a little bit more specific to the learning objectives. So that way we can kind of specifically point out, hey, these are the learning objectives. These are the specific things. But yeah, if you have any questions, comments, concerns, anything like that, please drop them in the comment. Leave a like on the video. Leave any comments, questions. I like to hop on and answer those as often as I can. So thank you so much. Have a great day.